Okay, so hello everyone. Thank you very much for being here today. My topic is TRP channels in the central nervous system. So first, let's talk a little bit about TRP channels in general, what they are. They are polymodally activated cation channels, meaning that they have a set of adequate stimuli that might activate them. And one good example of that would be the paired sensation of heat and spiciness. So I'm pretty sure most of you have already experienced this, that whenever we eat something spicy, we automatically detect it as if it was hot too. And that is the reason why, because it's the same ion channel that transmits information concerning both, namely the TRPV1 channel. So clearly, these channels are crucial for sensory processes such as vision, taste, temperature, sensation, and nociception. And since they are cation channels, they have a huge impact on membrane depolarization. Now, concerning the current scope of this presentation, we're going to discuss two TRPM channels in details. TRPM stands for myostatin here. In general, these TRPM channels are calcium channels, with the exception of TRPM4 and 5, which are impermeable to calcium. Now, this role in the peripheral nervous system of the channels is pretty well understood, but their existence and function in the central nervous system is not so well discovered. And one good reason for that would be that there's a lack of selective antibodies. So clearly, if we want to find these channels, we need new techniques for that. And first of all, to localize these channels, we used RNA-scope in situ hybridization. Now, what is this? This is a very sensitive and specific technique to detect even very small amounts of mRNA transcripts. And as you can see in this example here, for example, the TRPM4 channel mRNA transcripts are shown in green. And the picture is co-stained with SEDB1 that stains most cells. We'll talk about them a little bit later on. But the main point here is that, as you can see, that these TRPM4 mRNAs are transcribed and expressed later on the channels by these SEDB1 stained MOSI cells. So the two TRPM channels I mentioned are going to be TRPM4 and TRPM3. So let's see a little bit about them. First, concerning TRPM4. And also, it's important to note that besides TRP channels, we mostly deal with temporal lobe epilepsy. So most of the images that you're going to see here are from the medial temporal lobe. Namely, these are from the dentate gyres. And this might be familiar because this is what I just showed you a second ago. But if you look at the merged picture here, once again, the TRPM4 transcripts are shown in green, and the red staining goes for the MOSI cells. So there's a SEDB1 stained. Now, concerning TRPM3, our region of interest was the lateral amygdala. And what we found here was that it was mostly expressed by the principal cells, which are the excitatory cells in the region. And a little bit in details concerning what we had known prior to our experiments and what we know now. So first, with respect to TRPM3, it was first discovered as a noxious heat sensor besides the other members of the family, TRPV1 and TRPA1. Now, once again, this is a well-understood role of the channel, and there's already a compound used by Biohaven that aims at helping people with chronic pain sensation, especially in association with migraines. And by the way, this compound has already passed phase 1 trial, which is pretty promising. But according to more recent data, two de novo gain-of-function heterozygous mutations were found in the gene encoding for TRPM3, which led to the development of DEE in patients. That would be developmental and epileptic encephalopathies. And clearly, this rather complex syndrome includes a lot of epileptic activity and epileptic situations. But before we jump into epilepsy research and how we do it, it is an important side note that the lateral amygdala has long been known to be very much crucial to fear memory conditioning and fear memory encoding in general. So this is a little insight into our behavioral studies, which you're going to see here. So what happens here is we use wild-type and TRPM3 knockout mice, meaning that they do not have the TRPM3 channel. And you have the animal, and you co-introduce a painful and a non-painful stimulus to the animal. It could be a foot shock and a bell sound, for example. And this all happens in a given environment. For example, there's a distinct pattern on the wall or on the floor. This is crucial for later on memory retrieval, clearly. So upon the painful stimulus, you would want the animal to exhibit a sort of fearful behavior that is freezing in the case of mice. So as you can see, in the first case, there was no significant difference between the wild-type and the TRPM3 knockout mice, which is good in a sense that there was no baseline difference between the two groups. But upon the next set of experiments, when the animals were placed back into this given environment, you could already see a difference that the white-type animals displayed a significantly higher freezing tendency. Just as during the apex of these experiments, when you only apply the non-painful stimulus, and also the non-painful stimulus makes the animal exhibit a sort of fearful response, namely the freezing. So we could see that TRPM3 is also pretty much involved in managing fear memory encoding. But back to epilepsy. So the lateral amygdala is pretty much known to be involved in epilepsy as well. In many cases, it is the focal point of seizures, meaning that the seizures start from there, so they originate there. And also, if it's not the case, there are distinct anatomical and functional changes that appear with the amygdala involvement. That could be amygdala hypertrophy or sclerosis. And considering the functional consequences, for example, prolonged post-ictal confusion as described by patients. So altogether, the amygdala is pretty much involved in epilepsy. So about epilepsy research, this first of all is a sort of pool concerning the experiments that we generally do. Some of them we've already touched upon, for example, the RNA-scope in situ hybridization, the behavioral experiments. We also do in vitro electrophysiology. We'll talk about histology a little bit later on. And in vivo electrophysiology is highlighted because this is what I do mostly on a daily basis. So this is a scheme here. So first of all, since you want to record electric activities, you need electrodes, right? So we manufacture these electrodes, as you can see in the middle picture there, 100% DIY. And then these electrodes are implanted during a stereotaxic surgery, into target regions. That depends on the type of experiment that you want to do. And during this surgery, you can also apply different compounds depending on what you need, antagonist, agonist, whatever. And importantly, you can also apply cyanuric acid, which is a crucial point, because that is how we induce seizures in the animals. So as the animals come out of this anesthesia, they undergo a 1.5-hour EEG recording. That is to detect immediate seizures, the so-called status epilepticus. And after that, the animals might be used later on for histological purposes, they might be used for behavioral studies, or they might be kept for 24-hour EEG recordings, which is pretty crucial because this is how you can monitor if there is actual epilepsy developing in the animals or not. So if everything goes according to plan, you end up with something like this. So this is a 1.5-hour recording stacked together. That's why it's so densely packed. And as you can see, the white-type recording is significantly higher in frequency as opposed to the TRPM3 knockout recording. Also, there are other features of the recording that we look out for example, the interspike interval or total seizure time. These are also very informative to check. So far, based on our data, genetical ablation of the channel seems to be beneficial in the case of decreasing seizure severity. And concerning TRPM4, so as I mentioned, it's an interesting channel because it's impermeable to calcium. The reason why we started to work with this is that prior to our experiments, there had been information about the inhibition of TRPM4, how it is beneficial in the case of calcium-dependent pathologies such as arrhythmias, as you can see here. And I also mentioned that these TRPM4 channels are found in the muscle cells. Now, their function is not 100% understood yet. Our understanding so far is that they might stand as a sort of amplifier in the region, which is pretty important because this is a way how you can fine-tune incoming information and thus forward it to other hippocampal regions. But once again, it's not 100% clear yet. Now, coming to histology. So once you induce seizures, it is also pretty important to see how this all relates to cell loss and cell survival rates. So what you see here, once again, SETB1 staining. After the cyanuric acid injection, you can see that there was a significantly higher survival rate in the case of TRPM4 mice. So that was a pretty promising finding for us. And this would be an example of the spontaneous seizure detection, as I mentioned, the 24-hour recordings. Here, once again, TRPM4 knockout mice did significantly better with respect to decreasing seizure severity in these parameters. So yeah, the genetical ablation seems to be beneficial, but of course it's also pretty crucial to see how this might work on a pharmacological level. So for that, we're currently using meclofenamide. Wheels are in motion, but the data seem to be very promising so far. So now that I talked a lot about a lot of things, I would like to summarize this little presentation. The take-home messages here would be that TRP channels mediate several well-known processes in the peripheral nervous system, but there is increasing data that suggests that they might have a crucial role in the central nervous system as well. And highlighting TRPM3 and TRPM4, they seem to be linked to epileptogenesis, especially in association with temporal lobe epilepsy. Thus, they seem to be potential drug targets in the future. But as with everything else, further research is definitely needed. So there's nothing else left than the thank you page. So a special thanks go to the GOAT team. They are a wonderful group of scientists, and I'm really, really happy to be able to work with them. And I also would like to thank your time and attention with some blue-footed boobies. So thank you a lot.
Thank you very much. Congratulations, and please receive your certificate.